In this video, we are going to finish up the concepts of the introduction for biomolecules. Now, we introduced in the previous video the reaction of saponification. And remember that saponification is going to be the base hydrolysis of a triglyceride. And remember that in this base hydrolysis, the products for the reaction are going to be glycerol and 3 carboxylate ions. Now, you can envision that at the ester point, for each one of the acyl groups that are part of the triglyceride, the following reaction is going to happen. So when we are doing saponification of triglyceride, the following steps are happening at each one of the ester centers. So you can envision that this is going to happen 3 times. So what's going to happen is that ester motif, in which you can envision that glycerol is in this portion of the molecule, and here's where your fatty acid is. The hydroxide ion is going to first attack the carbon in the carbonyl and push the electrons up to the oxygen. In the next step of the reaction, we're going to have the electrons from the oxygen that used to be part of the carbonyl reform the C double bond O, and then it's going to kick out the oxygen that is part of the glycerol system. Once that oxygen has been produced, because we are producing a carboxylic acid, then the O minus can go and deprotonate the carboxylic acid, and that's how we form the carboxylate ion portion of the hydrolyzed triglyceride, and then the OH is going to be part of the glycerol system. Now, understand that when it comes to triglycerides, they do not need to have the same fatty acid at each position. We can have mixed triglycerides, and as you can see in the following example, this triglyceride is going to have 3 different fatty acids present. So as you can see, the top one is going to be palmitic acid, which if we classify it, that's going to be a saturated fatty acid. Then the middle one is going to be stearic acid, which is another saturated fatty acid. And the last one that we have present, that is the fatty acid that is coming from oleic acid, and you can see that that is an unsaturated fatty acid. So going back to the concepts of soaps, and I mentioned this briefly, when it comes to a soap, they consist of a fatty acid that it is now a carboxylate ion, okay? And remember, once hydrolysis occurs, you can see how that soap is going to have a polar group, and you can see that that portion is known as a hydrophilic portion of the molecule, because hydrophilic stands for a portion of the molecule that is going to be in love with water. That's what hydrophilic means. In love just means because it can interact with it because they have similar polarities. Now, the portion of the molecule that is where we have our carbon-carbon or carbon-carbon single bonds or carbon-carbon double bonds, that is going to be a hydrophobic portion of the molecule. That means that it is in essence afraid of water. But you can imagine that it is afraid of water because it's non-polar, so the polarities do not match. Now, One of the things that it is observed that whenever we are dealing with biochemistry, we at times don't draw all of these atoms together. We typically use cartoons. So if I label what would be the cartoon that I will draw for a soap, the polar group present in my carboxylate ion is going to be drawn as a circle. Then the non-polar side that you can see I'm circling in blue is drawn as a tail. So this drawing represents the structure of soap. Now, understand that these carboxylate ions are going to form micelles around the nonpolar particles. So this is what I was meaning with the soil that we're trying to remove whenever we are washing things with soaps. And then because of the arrangement of the molecules itself, that's how these nonpolar particles or soils are removed from the system. So you may be asking yourself, what is a micelle? Micelles are going to be a structure that is circular. And if we have, I'm just going to do a drawing about this. So if we have the soil or the nonpolar particles that we're trying to clean, when we combine it with a soap, what happens is that in essence, that soil, because it's nonpolar, is going to be interacting with the nonpolar tails of our soap. Remember that the circle is going to be representing the polar side of our soap. So once the micelle is created, then understand what is occurring overall. It's just that then the polar side will be able to interact with the water molecule. And that's why the micelles are going to be disposing of that non-polar particle that we are trying to clean up. Next, we are going to talk about phospholipids. And when it comes to phospholipids, understand that overall, these phospholipids are ester-like derivatives of phosphoric acid. Okay, so understand that when it comes to a phospholipid, which is the molecule that we have in the bottom, we are going to have two fatty acid components and understand that that phosphoric portion in our phosphoglyceride or acylglycerides, then it can be ionized and we can actually have functional groups be added to that phosphate that is in the bottom. So the reactivity here is going to happen at that phosphate. Notice that we have two esters only in the phosphoglycerides because we're going to have only two fatty acid components. Now, understand that the most abundant of the phosphoglycerides is going to be phosphoric diesters, as I explained previously. So when it comes to these phospholipids, we are going to have only two fatty acid components in them. Now, that last phosphate, as we're going to see, can have different groups added to it. Now, when it comes to phospholipids, understand that these structures are actually very important. So, a phospholipid, we can see the example here in the molecule of the left. Because what happens in the phospholipid is that we have, when it comes to the ester functional groups, 1, 2, and then the last one is going to be a phosphoester bond. And understand that for a phospholipid, when we have them in as a biomolecule, it's going to be bonded to an amino alcohol. Okay, and this amino alcohol, as you can see here, this is just an example of a primary amine. Now, this whole molecule is known as a cephalin. You don't need to know the different names that we have for the phospholipids. The important thing to understand is that when it comes to specifically the phospholipids, and remember that I mentioned that in biochemistry we actually do drawings, because drawing all these molecules out may be a little more of complexity. When we identify how these phospholipids relate to the molecule that we have on the right, I'm going to try to do justice in the color coding. Those green balls that you see here are going to be this portion of the molecule. So if I take one of these, this is just the portion of the amino alcohol. The two dangling thingies that we see here, this corresponds to the two fatty acids. Okay, so how we draw a phospholipid in essence is the polar portion with the two tails. And remember, the two tails are going to be the tails of the fatty acids. They don't need to be specifically saturated. This is just the figure that it is in the textbook, but it is important to note that the polar head is our non-polar tails. The polar head is the portion that has the phosphate and the amino alcohol. Then the non-polar tails are going to be specifically where we have the fatty acid portions of the molecule. Why are these structures important? Well, because in cells, even though this is an organic chemistry course, understand that this represents a structure in cells. So for those of you that have taken biology, this is called the lipid bilayer. And the lipid bilayer specifically is going to be the structure found in cell membranes. As you can see, we can envision that in this cell membrane, and the role in the cell is actually to keep the inside of the cell from the environment. So we can envision that here, those polar or those green circles that are representing the polar head are going to be interacting with substances that are polar, like water, and then the tails are meeting one another because they have similar polarity. So understand that our cell membranes in our cell have that structure. Now we are going to proceed to talk about the last members of lipids, and remember that these are going to be the lipids that are simple lipids because they cannot be hydrolyzed. For the first member of this group that we're going to talk about is steroids. So understand that steroids mainly function as chemical messengers. In order for something to be considered a steroid, they need to have this skeleton. You do not have to memorize the numbering of the positions in the steroid. Just know that this cyclic system, that is a 6-member ring, 6-member ring, 6-member ring, 5-member ring, this is called a steroid nucleus. And then this steroid nucleus is what determines if a particular molecule is known as a steroid. Understand that these ring systems must be bonded in that matter in order to something be considered a steroid. Now, you also have on letter A how it looks in terms of chair structures. And you can see that there's different diagrams for them. The main thing that you have to remember is the shape of that steroid nucleus, and that steroids are rigid, and the trans-ring fusions are the ones that are going to be preventing this molecule specifically from ring flipping. Now, you may be asking yourself, where do we see steroids? Well, steroids are found in many molecules inside the body, but one of the examples in which we see this steroidal molecules is in sex hormones. Okay, so sex hormones are going to be these biomolecules that are released at a particular time during development, and they are going to be regulating tissue growth and reproduction processes. Now, when it comes to steroids, if we compared male sex hormones and female sex hormones in terms of the structure, we can really see that steroidal or that steroid nucleus present in the molecule. I'm just highlighting them in pink. That's how you know that these molecules are known as steroids, because you can observe the cyclic system that I pointed out specifically a few moments ago. Now, understand that male hormones are known as androgens, and female hormones are known as estrogens or progestins. This is just illustrating their names for them. For me, it's very important that you can look at a molecule and identify that it is a steroid. And the ones that we have here are just 2 examples of androgens. Now, if we look at the molecules for estrogens and progestin, which are the female sex hormones, we can see again the cyclic systems. Now, you may notice. Now, I'm just going to highlight the one that are present in estradiol. Specifically, the ring system don't need to be alkane base. As long as you have the correct number of carbons, it's going to be considered a steroid. Because if we look at the structure of estradiol or estrone, we can see that the first ring system is actually a benzene ring. Okay? Now, understand that when it comes to estrogen and progestins, they are the ones that are involved in ovulation during pregnancy. But the main thing that I want you guys to take into consideration is that if you look at a molecule, I can identify that out of the lipids, that's going to be specifically a steroid, because we have that steroidal nucleus that was highlighted in pink in the previous structure, in the previous slide, and in the following. Now, these hormones or this steroid hormones, because not all hormones are steroid based. The ones that I just illustrated are the ones that are steroid based. Understand that synthetic hormones, as you can see shown below, they are designed to mimic estrogen and progestins and to inhibit ovulation. So whenever a female decides to take birth control, understand that those birth control pills are going to have molecules that mimic the structures of those female sex hormones, and that's how ovulation is actually, or that's how ovulation is controlled, or the whole process of pregnancy. Now, when it comes to the last class of molecules that are known as lipids, understand that terpenes are going to be specific molecules that are isolated from plant oils. These terpenes are going to have a strong fragrance, okay? And understand that they're used as flavorants and odorants. So if you're one of those people that actually utilize essential oils in your everyday life, what you're utilizing is a terpene. To give you guys examples of these molecules, here we have menthol, which is isolated from peppermint oil. We have camphor, which is isolated from evergreen trees. And then here we have carbone, which is actually the flavor of spearmint. When it comes to the terpenes, understand that they can be classified by the number of carbons that are present in them. And as you can see in table 26.4, we have the different classification of terpenes. So if you have a terpene that only has 10 carbons, that is known as a monoterpene. If it has 15 carbons, that's a sesquiterpene. 20 carbons is a diterpene. 30 carbon atoms is a triterpene. 40 carbons is called as a tetraterpene. So for example, beta carotene is known as a tetraterpene, and beta carotene is going to be the molecule present in food like carrots, that gives its orange color. In some of the good properties that are obtained nutrition wise from carrots is because of beta carotene. And this concludes our talk about biomolecules.